Myself, Dr. Vijayanand, Associate Professor, Department of Life Sciences, Christy Jayanti College, Bangalore. So, topic for the today's lecture is biosynthesis of fatty acids, fatty acid biosynthesis. So, fatty acids, we know mainly they are synthesized. So, there are three main systems which are involved in the fatty acid biosynthesis. When you talk about biosynthesis of fatty acids, so which includes mitochondrial system, extra mitochondrial system and microsomal system. So mitochondrial system, so which is the elongation of the existing fatty acid, whereas when you consider the extra mitochondrial system is the widely accepted system for the biosynthesis of fatty acid, which is also called as de novo synthesis of fatty acids. So the third one is microsomal system. So again it is mainly meant for the extension of the medium chain fatty acids. So today's lecture we are going to talk about the biosynthesis of fatty acid. So mainly de novo synthesis of fatty acid biosynthesis. So fatty acid biosynthesis we also call it as lipogenesis, is nothing but synthesis of fatty acids, where fatty acids are mainly synthesized especially in the liver. So and this de novo synthesis of fatty acids so mainly takes place from the acetyl CoA. So acetyl CoA is the starting substrate, so that takes place in that probably occurs in the cytosol. And the enzyme system, in this lecture we are going to talk about the enzyme system which are involved in the biosynthesis of fatty acids. So here, talking about this de novo synthesis of fatty acid, it mainly takes place in liver, kidney, to some extent it takes place in adipose tissue, lactating mammary glands. And the enzyme systems, when you talk about the enzyme complex, that is fatty acid synthase, are mainly located in cytosol. And this synthesis process or this system of fatty acid synthesis, we also call it as extra mitochondrial system or cytoplasmic fatty acid synthase system. And main source of this fatty acid is NADPH, so that provides the reducing equivalence, and this NADPH is mainly obtained from the HMP shunt, and every molecule of acetyl CoA delivered to the cytoplasm, one molecule of NADPH is formed and ATP supplies the energy. So that is how this is connected. So this pathway, when you talk about this pathway, so that is biosynthesis of fatty acid, there are three steps which are involved in the fatty acid biosynthesis. So the first step is production of acetyl CoA and NADPH, and the second stage is conversion of acetyl CoA to malonyl CoA, and then the further reactions that takes place which are mainly catalyzed by the fatty acid synthase complex. So we need to understand so mainly the sources of acetyl CoA as well as NADPH. So those are the important compounds, especially when you consider the biosynthesis of fatty acids. Acetyl CoA, as I mentioned, it is the starting substrate especially for the fatty acid biosynthesis. It is produced in the mitochondria, so by oxidation of pyruvate. So when pyruvate is getting oxidized, so pyruvate is oxidized by pyruvate dehydrogenase. So ultimately, so they are converted to, that means acetyl CoA. Then fatty acid degradation, so of carbon skeleton of certain amino acids and from ketone bodies also provides the acetyl CoA. So mitochondria which are not permeable to the acetyl CoA. So whereas here we need to understand how this acetyl CoA is transferred from mitochondria to cytosol. So if you talk about the cytosol, so probably this fatty acid biosynthesis will take place, and from mitochondria, so the oxidation of pyruvate to acetyl CoA, so which is enhanced mainly by pyruvate dehydrogenase complex. So after that, so this acetyl CoA, so it is a major substrate for the fatty acid, that means biosynthesis, and this NADPH is the major reducing equivalence compounds which is mainly utilized for the fatty acid biosynthesis. So this acetyl CoA, so which is also mainly obtained from the TCA cycle, which is also widely used as substrate for the biosynthesis of fatty acids. So here there are different steps which are involved in the fatty acid biosynthesis. So the first reaction is formation of malonyl CoA from acetyl-CoA. So the starting substrate is acetyl-CoA. So this acetyl-CoA is carboxylated to malonyl-CoA by the enzyme acetyl-CoA carboxylase. So this is an ATP dependent reaction, and this reaction requires biotin for CO2 fixation. So here this acetyl-CoA, so which is converted to malonyl-CoA, so which is catalyzed by acetyl-CoA carboxylase. So these are the enzymes which are involved in the synthesis of fatty acids, so which is a multi-enzyme complex which are collectively called as fatty acid synthase. So this fatty acid synthase is a dimer which consists of 6 enzymes and 1 acyl carrier protein. So the enzyme multi-enzyme complex, which includes ketoacyl synthase, acetyl transacylase, malonyl transacylase, dehydratase, enoyl reductase, ketoacyl reductase, thioesterase, and 1 acyl carrier protein, that is ACP. So this enzyme unit is active so when these 2 dimeric units are joined together. And this enzyme complex fatty acid synthase, so which is composed of 2 identical subunits, and the molecular weight is high, so almost 240,000 Daltons. And these subunits of these enzymes which play a vital role in the synthesis of fatty acid. And if you consider the reactions of the fatty acid synthesis, so the first step is the conversion of the acetyl-CoA to malonyl-CoA, so which is catalyzed by acetyl-CoA carboxylase. So here the carboxylation of acetyl-CoA, so that leads to form malonyl-CoA, and then this malonyl-CoA is converted to malonyl ACP. So here, so ACP transferase is mainly involved, so that it leaves CoA and it replaces the ACP. So that is how the malonyl-CoA is converted to malonyl ACP. So once malonyl ACP is formed, again this malonyl ACP condenses with one more molecule of acetyl-CoA. So here this reaction we call it as condensation reaction, which is catalyzed by synthase. So ketoacyl-CoA synthase is the enzyme, so that it results with formation of acetoacetyl ACP. So once this acetoacetyl ACP is formed, then it undergoes reduction reaction, and this reduction reaction utilizes NADPH for the reaction, so that acetoacetyl ACP is converted to hydroxybutyryl ACP. So hydroxybutyryl ACP undergoes dehydration. So when it undergoes dehydration, so it is converted to crotonyl ACP. So as a result of crotonyl ACP, again it undergoes one more round of reduction, which requires NADPH for the reaction. So ultimately the crotonyl ACP is converted to butyryl ACP. So after forming this butyryl ACP, so here it undergoes 6 cycles. So when the cycle is continued for 6 cycles, so especially it results with the addition of CH2 group, so that butyryl ACP is converted to palmitoyl ACP, and in the final reaction, so this butyryl ACP, so which is converted to palmitoyl ACP, and further it is catalyzed by, that is thioesterase, so which is involved in formation of palmitic acid. So that is how the fatty acids are formed, and the net energetics of the fatty acid biosynthesis, mainly it includes so the number of ATPs which are formed from the fatty acid oxidation. So here mainly the energy molecules, that is 8 acetyl-CoA molecules are being oxidized, so and 14 NADPH molecules are also involved, so that leads to form the palmitate. So ultimately it produces 14 NADP plus, and if you consider the overall reaction, so the acetyl-CoA, so when it condenses with malonyl-CoA, so with the presence of reducing substance, that is NADPH, so it results with formation of palmitate as well as NADP. So the palmitate which is formed over here, so which is considered to be so the ultimate fatty acid biosynthesis. So with this lecture, so the fatty acid biosynthesis is overview of fatty acid biosynthesis over. Thank you.